early next week. Okay, let's have a class. First of all, it's been very interesting. I did on my hands. We want to see the bacteria grow on my hands, what it looks like. See, I put this on my, remember I touched my hands here? Mannitol salt agar. See the big two blue one? That's modes, East Tender modes. Did you see the smaller one? The yellow one? That's very possible Staphylococcus because it's yellow color. Remember? It's mannitol fermentation positive. It will grow because it's like salt, so it's a halophile. Remember the question? So that's very possible from my hand has some Staphylococcus. Might not be ours, could be other Staphylococcus species. We will mention in the exam 2 lecture. So it looks like, you know, that's big one of most. Okay, now let's look at this blood agar. Well, blood agar have a big East Tender modes growing there. Look at the big one. These another big smear there. So creaming looks like it's grow a lot, dramatically. I don't know what is that. Okay, this is a Makanki agar. Looks like have some E. coli there, but not really pink color. So some gram-negative bacteria. Let me see this one. This is, we call it Campylobacter agar, because I don't have Campylobacter, so it looks okay. This is, I'm kind of a little bit surprised. This agar doesn't have anything grow. Looks like a TSA. Why? Because we add antibiotics there, which is natric acids. So the antibiotics will kill lots of the bacteria. So that's why this is a traditional tropic soil agar bloss, but it didn't have much growth. So it's very interesting because I have antibiotics there. The antibiotics will kill the bacteria from my hand. I did this one. This is also Campylobacter agar, doesn't have much. Because the Campylobacter usually have to grow in the anaerobic jaw, microallophilic jaw. So you're not gonna grow dramatically like that. So you can see that's verified on our hands. There is a Staphylococcus. And it's yellow color, so really good. And there is East Tender modes, obviously. Okay? So we will be moving on to our exam 2. I think most of you will be like the exam 2 better than exam 1 because we will talk about lots of the bacteria which is commonly associated with the human infectious disease. So we will talk about all of that. And the exam, we will talk about some case studies, and you're going to do the match which one is which. All of these are very common infectious disease happening in the United States, which is CDC recorded, has a longer history in the whole country. Now, first of all, we want to talk about the respiratory tract a little bit. So this is anatomy. If you never learned, we just show you a little bit. So you have esophagus here. You have a bronchus, lungs area, alveolar area. This is the lung area. Then very important here, you have a very soft bone, which is epiglottis. So remember, when you were very young, even in the kindergarten, your mom or your teacher will say, please do not laughing or talk during the lunch, because the epiglottis is a soft bone. When you eat, it should be closed. If you're eating and then you're talking or laughing, then the food will go to the lung area instead of go to the intestinal area, go to the stomach, so you could get a choke. So that's a soft bone there. Okay, that's the intersection usually between upper respiratory tract and the lower respiratory tract. Now, another thing I also want to mention is a tonsil. There's a tonsil area right here. I know some of you do not have a tonsil, or the tonsil has been taken out when you were very young. What's the reason? because tonsil is a very good reservoir for bacteria, even for microorganisms and also for virus. So lots of the people took out of the tonsil when you were young. Now, oral cavity and the nasal, this area, nasal tract, obviously there's a lots of the bacteria. But those bacteria basically stay there, which means they are localized. They should not cause a problem. But when the condition changed, they may translocate it from the upper respiratory tract to lower respiratory tract. Then they will be cause a problem. We call it the opportunistic pathogen, which means the bacteria looking for the opportunity to cause a problem. Okay, so just very briefly talk about the respiratory tract. We're not going to do the anatomy exam for you to label all these, but be careful about it when we talk about the bacteria, microorganism, tonsil area is a big one. Okay? Now next, we want to be introduce several of the bacteria started from today and relate to the upper respiratory tract disease. Then we move on to the stomach, intestinal areas, and we talk about the sexually transmitted disease later on. Okay, so the first one, which is right here, you see the name at the bottom is Corynebacteria diphtheria. Corynebacteria diphtheria. Okay, we review, we learned in the exam 1, this is genus name, this is species name. Now before we talk about this, we will be reviewing what we talk about for the gram stain. Remember, we talk about the gram stain, there is 3 information. Now, number 1, gram reaction, positive, negative. Number 2, which is morphology. And number 3 will be the arrangement. So we want to talk about the characteristics of all of these pathogens. We're going to be focused on that. So Corynebacteria diphtheria. Where is this name comes from? The corner. This is a Greek word, means club. Because the shape, the morphology of the bacteria and the arrangement of the bacteria means that. So at the diphtheria, this is the toxin name. So what is that? This is a gram-positive, large shape, and the arrangement is club. And if you look at the picture, the club, it looks like this. It looks like that. It could even looks like this. So different shape, but 2 of the people stacked together seems like. And usually inside, if you look very carefully, they should have a dots there. And their dots, we talk more details that called the inclusion, because they usually do storage of phosphates or the other chemicals. Okay, so there's a little bit of machinery inside. So it's gram-positive, large shape, large and the club shape, large shape and club arrangement. And by the way, this is under the phase contrast microscope, so it looks like that. Now, because of them, they stack together, so we also call it a palisade. We also call it like that. We also call it a palisade. Okay. Now, what is this one for? This is the one, the pathogen. Usually, it is for the immigrant kids to the United States. So why? Why we want to talk about that? Talk about the immigrant kids is a risk group. Because in the United States, we had a vaccine for that. The vaccine is called DTaP. This vaccine is one shot, and one shot, 3 jobs. So what is the D? This is diphtheria. What is the T? This is Clostridium tetanus. A, which means acellular, and the P is Bordetella pertussis. This we will mention later on these two. So that's one shot for 3 jobs. So Caulobacter diphtheria, which is covered by this vaccine, and if you look at the United States recommended immune scheduling, you require to have this one at 2 months, 4 months, 6 months, 8, 12, and you need to have a booster shot later on. So remember, when you were born, after about a month, or even 15 days, then your mom has to schedule, find a pediatrician. The pediatrician needs to establish an immune record for you. And you recommend to see the doctor every 2 months when you're less than 1 year old. Older than that is half a year. Then older than like 3 years old is every year. Then the doctor, your pediatrician, will be go ahead to see whether you are covered by those type of vaccine shots. And DTaP is that. So anybody who is natural born in the United States, they require you to take these shots. And you do require to do. Otherwise, you go daycare or even go elementary school, you get restricted. So in the United States, this is not a problem, but for immigrant kids, usually it's a risk group. Okay, what type of the symptoms it will have? It's very interesting. The symptoms what you have is, so let's say a human being, you look at that, okay, let's draw a human being. Briefly, this here, you will have a very sick neck. And this sick neck usually accompanied with lymph nodes swelling. That is we call like a ball, because it's a ball neck. So that's the first symptom. Half of these neck are really swelling. And then, if you see the tonsil area, let's say look at here, the tonsil area, you will see a very typical symptom, which is we call acidomembrane. It is kind of like soft, weak, and if you touch it, using cotton swab to touch it, it will be bleeding. And then the kids will go wow, wow, wow by the time. And then the doctor will say, did you have a vaccination? And usually people will say, we didn't have a vaccination. So that's acidomembrane, the tonsil area, and this ball neck usually is a typical symptoms for Caulobacter diphtheria. Now, we want to mention about this is this pathogen is localized. Localized in tonsil, which means they stay there. They do not be translocated to other places. Don't go stomach, don't go intestinal. However, the toxin, they will be transferred. So we call it relatively invasive. Now, what is the toxin? The toxin is diphtheria toxin. We call it an exotoxin. Now, where is that toxin comes from? This is a theory we talk about. Yes, long time ago, there is a bacteria. Okay, we have a bacteria genome. We have a bacteria genome. Then we have a phage. It's a virus. This phage we call it a beta phage. And this phage usually is a virus. Okay, they could attacking a bacteria. Then this virus, this phage got attacking a bacteria, they carry on diphtheria toxin. So what happened is that they will be copied and also inserted their genome, the toxin genome, the sequencing, into the bacterial genome. And they stay there, very similar like a parasite to host. Very similar. And they will stay there forever. Become part of the bacterial genome, will be translated, transcribed, translated, become a toxin. And this kind of the phenomenon, we call it a lyse journey conversion. So it's very interesting. It's one of the theory why bacteria will be carry on those toxins. So you learned in your other classes already. So for example, why eukaryotes have a mitochondria, have a Golgi body, have a ribosome, all those things. They think a long time ago, there is a small bacteria or a small virus which have those machinery, and they eat it by a fungi and become a part of their body. Or sometimes they integrate the genome and express those machinery. That is called endocytosis, is that right? And this is lyse journey conversion. The theory is very similar to that. So they think why this bacteria has a toxin, because a long time ago, a bacterial phage, a virus has a toxin. And the phage is attacking a bacteria, but they did not kill the bacteria, and they are integrating their DNA sequencing in there. And like a parasite to a host, and the toxin is there, and the toxin plays a role. So what type of the role the toxin going to play? We could look at this here, the picture right here, okay? This is the one we usually call, it's an AB toxin. So they have two parts. And then when they go inside of the body, the AB toxin will generate, we call it an indocin. And usually the environment, the pH is less than 5.0, so it's a very acid environment. And then the AB toxin will be released to do their body. So A toxin, we call it an A toxin domain. So A domain, it's a toxin domain. Generate the toxin. And B domain, actually, they will release. You can see the picture there. This is a transmembrane domain. So transmembrane domain, what's the job they do? Think about on the skin surfaces. In utensil area, something going to be transferred to it. What's going to happen? They're going to cause bleeding. That's why the cell membrane is there, is soft and weak. You touch it, it's going to start bleeding. Now, what's the toxin domain going to be? They will have inflammation. So that's what these two things will be work together to do. Now, if you want to do a little bit, digging a little bit further, what does toxin really do? This will prevent host cell, the RNA translation, because they will be attacking elongation factor 2 during the translation of the RNA. So just very briefly, we talk about the mechanism behind it, okay? This pathogen, if you want to really do something for the treatment, honestly to say, it is difficult to do. If you already have it, we have to using antimicrobials. Not really the antimicrobials. Antimicrobials in the medical area, we call it antibiotics. So such as like penicillin, methicillin, all those things. But more important, because this is a toxin, so you also have to use an antitoxin to neutralize this toxin first. So those two chemicals, you have to do both. But more important is this, is very important is this vaccine. Because that vaccine, if you do, it will prevent these diseases. So you always remember, even a bacterial cell, even a virus, it is more important for the prevention is to do a good job of the vaccine. Although we have a debate about that, but the vaccine is always recognized is a most effective way to prevent the infectious disease related compared to you do some treatment afterwards. And I will be telling you later on when we talk about antibiotics. Most of the antibiotics, it will work very well, is for gram-positive bacteria. Do you know why? It's a very simple reason. They have a target is so obvious, it's peptidoglycan. And the gram-negative bacteria, it does not work very well. So I would give you a very simple example. You have a Streptococcus pneumoniae, Streptococcus pyogenes. If you have a strep throat, you go to see a doctor, doctor will give you antibiotics. It works well. But you heard about all of those outbreaks of the foodborne pathogen, is that right? E. coli, salmonella. Why the people will die? Well, of course, they go to the hospital, it's already late because it's onset time. Another thing is that those are gram-negative bacteria. So gram-negative bacteria, honestly, you're using antibiotics doesn't work too well because their cell wall structure, the peptidoglycan is very thin. Another thing I also want to mention is even you use antibiotics for gram-positive bacteria, the more effective is when the bacteria is growing because they are building the peptidoglycan. So you're attacking it, then it will stop the building, will be more effective. If the bacteria is already there, you're using antibiotics, that takes way longer time to do. So there's something tricks behind these treatments. So the timing is very important. Okay. Now, this pathogen, usually if you had it, what do you have to do? You do need to take the cotton swab from the tonsil area, you put it. You need to go to a transport medium. The transport medium needs to go to reference lab, CDC reference lab, to report it and to confirm it. And the transport medium here, we call it, the full name, we call it a cysteine telluride blood agar. It is a cysteine telluride blood agar. And when it's on cysteine telluride blood agar, it is black color. Yes, you have to do that. Cysteine. If you know amino acids, CH2, SH. Is that right? It's a sulfide. The amino acid that has a sulfide. It's a black carbon. So the transport media, you need to do that. So this is what I talk about the Corynebacterial diphtheria. So I hope you want you going to keep the recording of these characteristics in a flashcard if possible. And then later on we have case studies, and we will give you a little bit of story which is coming from the CDC recording long time ago. Then we will ask you what's the possibility of the pathological pathogen it is. So you need to tell me it is Corynebacterial diphtheria. Okay. Second one we want to talk about is meningitis. This happens very often in the United States. Seems like not many good pandemic. The next one is meningitis. Meningitis, first of all, this is a little bit of brain membrane system disease. It's inflammation between brain membrane and the cerebral spinal cord. It's an inflammation between that. The symptom of the meningitis, usually we have a confusion, lethargy, sleepy, you may have a vomiting, and you have a photophobia, scared to the light, and very important, people doesn't recognize it, tough neck. This is people usually doesn't really pay much attention. They think the tough neck usually is a muscle problem, but if you have a tough neck, if you go ahead to sleeping, take a rest for like 2 weeks, still doesn't, not even a week it doesn't recover very well. Be careful, there's maybe meningitis. And I forgot to mention about the Corynebacterial diphtheria. This toxin is also a neurological toxin. It is a neurological toxin. This toxin will cause double vision accompanied with difficult speak and difficult swallowing. Which means you're eating something, you may have a problem, okay? Now, meningitis, the pathogen caused the meningitis, it is relatively, we say, it is age dependent. If you're a newborn baby, 0 to 2 months, the pathogen, we call it GBS. It is called Group B, Streptococcus agalactiae. We will mention detail later on what is Group B, what is Streptococcus agalactiae. If you are 2 months to 5-year-old very young kids, the pathogen responsible for it called HIV. What is that? Type B Haemophilus influenza. This is, we will mention real quick. It's a fastidious bacteria. It is the #1 childhood pathogen. If you are older people or adults, 5-year-old to 40-year-old, it is Neisseria meningitis. Neisseria meningitis. If you're older people, more than 40 years old, it is Streptococcus pneumoniae. Very interesting. It causes ammonia, but actually for older people, it will cause meningitis. Medical area, more than 40 years old, we call it older people. I'm 43, 44. I'm older people compared to you guys, and you guys are treated as adults. Okay, that's a medical definition. So, not go 60 years or 70 years, more than 40 years old, you're older people. So, that's age dependent. So, we want to talk in detail of these one by one. It's very interesting. Lots of the information will be tell you. And this slide tells you vomiting, headache, lethargy, confusion, but the tough neck. So we want to mention this guy first, is Haemophilus influenza. That's the first one we want to mention. This is a very interesting name. Influenza. When you hear influenza, very interesting. You would think about it is kind of like a flu-like symptoms. That's called influenza. Avian influenza. You heard all the time. You have a coughing, sneezing, fever. However, it comes like cause meningitis, because the first time when people isolated Haemophilus influenza, it do coming from the people who had mild symptoms, but later on they find it's flu-like symptoms. Later on they find it's related to the meningitis. This pathogen is gram-negative Coccobacillus. What means Coccobacillus? It's kind of like this. Very short rods. Looks like a cocktail, looks like a bacillus. So it's a rod shape and a spiral shape, a circle shape between that. So we call it gram-negative Coccobacillus. Obviously, it's a single. Okay, it's a single arrangement. This pathogen is a fastidious bacteria. This is related to what we learned in our exam 1 last week. What means fastidious bacteria? We talk about the bacteria agar. There are 3 major agar using in the clinical area for isolation, identification bacteria, which is McConkey agar. That's why we want to talk in the class. McConkey agar, blood agar, and chocolate agar. If we say this pathogen is a fastidious bacteria, what this means? It doesn't grow on the McConkey agar. It does not grow on the blood agar. It only grows on chocolate agar. That's the first thing you need to know. Another thing, this needs a very special ingredients. What happened is, people did a research before. This is a traditional TSA agar, tropic soil agar. Okay, I'm going to add in the Haemophilus influenza. It's not going to grow. We know it's a fastidious bacteria. However, if I'm going to do something special, we put an X factor there. We put a V factor there. It didn't grow, but we add X and V factor together, the bacteria grows very well. What is X? It's hemi. What is a V? NAD. What is that? Nicotinic adenine dinucleotides. It's an electron carrier. They need a very special ingredients to grow on the tropic soil. Now, more than that, it is very interesting results. If I have agar plates, because very interestingly in the clinical area, we're using blood agar plates. Okay, we're using a blood agar to do the identification. We want to see the hemolytic, beta, alpha, hemolyzing. What happened is, it's not going to grow because it's a fastidious bacteria. However, if I'm going to grow Staphylococcus aureus, it's very interesting. I inoculate Haemophilus influenza, they're going to show up with these tiny dots. So we call this is a satellite phenomenon. That's a satellite phenomenon. So you can see, it is also another ironic pathogen. Is that right? We call this is #1 childhood disease pathogen in the United States called meningitis. But what happened, it didn't grow on the TSA agar, neither Hemming and NAD as a supplements. We call it XV factor. And it didn't grow on the blood agar. It needed Staphylococcus aureus to generate the X and V factor, because blood agar, there is a Hemming there already. And the Staphylococcus aureus growing there will create NAD. That's why there is this satellite phenomenon comes. And only chocolate agar it is grown. However, it's caused a big problem in the United States. History. Okay, right now you had a vaccine, so it's not a big deal. So it's very interesting. Now this slide here, which is showing you several pictures. Haemophilus influenza grow on the chocolate agar. What kind of brownish chocolate agar looks like chocolate? Really good. XLV factor, you see, X factor didn't grow, V factor didn't grow. Only when they combine together, you see the circle, the shinish color there, that means the pathogens grow. So last one, this is blood agar. If you streak Staphylococcus aureus there, you see very tiny colony there. Look at how tiny these colonies grow. That is a satellite phenomenon. Okay? So it's interesting for this passage. So we talk about this guy is Haemophilus influenza. Okay, next one we want to talk about is Neisseria meningitis. We want to mention this because this one related to the all of you guys. This is called Neisseria meningitis. Meningitis. Okay, Neisseria meningitis. It is the same as same genus name as sexually transmitted disease called Neisseria gonorrhea. Same thing, very similar, but one causes gonorrhea, one causes meningitis, and which means Neisseria. Okay, this pathogen is also gram-negative. Bacillus, but it's very interesting. It's diprococcal. It is coffee bean shape. What it looks like? Coffee bean. Sorry, not this shape, not this shape. Like this. Coffee bean. Looks like that. So the old time, when you have a coffee bean, you have a coffee machine, you have to crush it, is that right? It makes a powder. That's coffee bean. And then when you look at here, you see those red one, coffee bean shape. That is surrounded by the white blood cell. That's what the picture looks like. Okay? It's gram-negative bacillus, coffee bean shape. It's Neisseria meningitis. This one in the United States, they have a vaccine, and they cover limited amount of the serotyping. The serotyping is A, B, C, W, 135, and Y. It is very interesting. In the United States, the serological type of being is responsible for 60%. The risk group undergraduate in the college. And I want to tell you a story. There's a true story happened in Indiana University. A boy called Ryan, and he had a little bit of fever, like sneezing, like those mild symptoms like influenza, or flu-like symptoms. And he had a very special happen, is a tough neck. So he think he can take a break just by sleeping. So he's sleeping in the dorm for 1 day. They're not feeling really recovered, and sleeping another 24 hours, and eating a little bit, and finally a little bit dehydration, and his roommate took him to the Indiana University in Bloomington and the medical center. And the doctor goes there to give the antibiotics immediately. And the doctor said, if you come here another hour later, you're going to be dying. This tells you, undergraduate in the dorm or in the library or in the very crowded area, like in the bar, it's a big risk. So risk group is undergraduates. And the guy, Ryan, which is saved by the medical doctor in the school, and he's really encouraged. He's a biology student, so later on he's going to study further for the meningitis. And after graduate, he works at the CDC for some of the foundation to give the donation from the other people to encourage the treatments for the meningitis. So that's give you a story which is telling you 60% of them for the undergraduates, and it's a type of B. It's a type of B serological typing. Now, in the history of the United States, before 2010, they used to have a big debate. The vaccine there, you have MPSV4, MCV4. Those, we call it monoamine, the commercial name. They only cover 40% because they cover A, C, Y, and W135. They do not cover the B. But a lot of the states, they require the undergraduates to take in this vaccine allowed to register for the class. It's in California, Connecticut, and Massachusetts, or those places. Then there's a big debate for a long time, because people will say, you had the vaccine is only cover 40%. There is a 60% of the chance people are still going to get it. Why you require people to do it? So the science area works very hard until 2017 to 2018, not a while ago. Right before the pandemic, they have a new vaccine comes out, which is could cover the B. So right now, this is a problem, not a problem anymore. Okay, so it's very interesting. I just want to let you know this meningitis thing happens in undergraduate, the coffee bean shaped, it is a big risk. Now, usually what happens is when you go to see a doctor, the doctor see you have a lethargy, have a confusion, scared to the light, a little bit sleepy, not much energy there. They will give you is a CSF, cerebral spinal fluid. Cerebral spinal fluid. They will get a sample from there, and then they usually very quickly they could do a gram stain. So you're going to see the morphology, you see the gram results, morphology, and the shape. You give a decision. Now, for Neisseria meningitis, this is also a fastidious bacteria. The good thing, these are fastidious bacteria, so this is only on chocolate agar. And not only on a chocolate agar, it's only on modified chocolate agar, which is called modified Sayre-Martin medium. It is actually a very special medium that they grow it. So very simple, you have a spinal fluid, I do the gram stain, I see the coffee bean shape. And I streak onto blood agar, I didn't see grow. Streak onto Macanthi agar, I didn't see grow. I need to use chocolate agar. Well, 90% it's Neisseria gonorrhea. Another thing we will be mentioned briefly, this is oxidase test positive. which means using oxidase strip, it turns blue color. So what is oxidase? You know the NADH2 go to FAD, go to FMN, FES, that is the electron transport chain, go to coenzyme Q, go to cytochrome C, then go to cytochrome A, go to A3, and finally go to the oxygen. Is that right? There is an electron going to be transferred. Now, once the electron transfer go to the cytochrome C, they need some of the enzyme. This enzyme called oxidase. So oxidase, we usually call it a cytochrome C oxidase. And it's a very special test. The oxidase test is positive. So, which means you do a simple strip testing, use oxidase. You dip it there, it turns blue brownish color. A little bit brownish, relatively blue purple color, I should say. And that's very possible Neisseria meningitis, and also the age group, like I just mentioned. This will be, if you are like 25 years old, 20 years old, fit in this group, then bingo, that's Neisseria meningitis. So, kind of interesting with these things. Okay. Next one we want to mention is, by the way, this is another good example for coffee bean shape. Okay, that's another very good example for that. Pertussis. Pertussis, what happened? This is the one, I want to remove here. This is the one actually covered by DTAP for the Bordetella pertussis. Bordetella pertussis. Now, first of all, this is covered by DTAP. This means pertussis. Pertussis is very interesting. This is happened